Welcome to excretory products and their elimination chapter. Now we will see in it human excretory system. Till now we had seen what are the nitrogenous excretion products, ammonia, urea and uric acid, and in which organisms they excrete. This is the structure of human excretory system. System is made up of different parts like kidneys. This is kidney, 1 and 2 kidneys are there. Then ureters. This pipeline structure is ureter. This is urinary bladder. This bag like structure is urinary bladder where the urine is stored. Human excretory system consists of a pair of kidneys, 2 kidneys, a pair of ureter, 2 ureters, a urinary bladder, 1 urinary bladder, and 1 urethra. Urethra means from where the urine is out of the body. Here in figure, adrenal glands, where these glands are above the kidney. This is inferior vena cava. This is aorta. This kidney open they have showed. Here it is pelvis where all the filtered kidney comes, and from here through ureter it is sent to bladder and through urethra out. This is cortex, medulla. Cortex is outer, medulla is inner part. Now we will see one by one the organs. The first, the kidneys, which are the main part of excretion. These are reddish brown like bean shaped structure situated between the levels of last thoracic and third lumbar vertebrae. If you stand like Vittal, keeping your hands on your waist, where your fingers point, their kidneys are situated beneath it. These are close to the dorsal inner wall of abdominal cavity. Each kidney of an adult human measures 10 to 12 centimeter in length. How much? 10 to 12 centimeter, and width 5 to 7 centimeter, and 2 to 3 centimeter in thickness, with an average of 120 to 170 gram. That is 150 grams in males and 135 grams in females approximate. Kidneys are mesodermal in origin. They develop from mesodermal nephrostomes or mesomeres, ciliated structure functional in embryonic conditions. Mesodermal means there is one part in the embryo. When the embryo or the baby is forming in the womb of an mother, this mesodermal part, they form kidneys, or they have the origin of kidney cells. Now this position of kidneys, the kidneys are located below the diaphragm on the left and right sides. The right kidney is lower and smaller than the left kidney because the liver takes up much space of the right side. Now we will see structure of kidney. Structure of kidney can be studied under two headings, that is external as well as internal structure. Now we will see external structure. The outer surface of each kidney is convex. Okay. Inner concave surface has a notch like hilum. The supply of blood occurs through hilum, that is renal artery and renal vein, pass in and out of kidney along with the ureter and the nerve supply of kidney. This is hilum, this concave part. From here renal artery, renal vein and ureter pass. If we look from outside to inside, 3 layers cover the kidney, that is renal fascia outermost, the adipose layer, and then renal capsule, the innermost layer. These coverings protect the kidneys from external shocks and injuries. Now, this external structure consists of renal fascia, adipose layer, and renal capsule. Convex and concave surfaces. Convex outer, convex inner. Inner is also called as hilum, from where renal vein, artery, and ureter pass. Now we will see internal structure. Now the section of a mammalian kidney shows an outer cortex and inner medulla. Inside the kidney, the ureter is expanded as a funnel-shaped cavity called renal pelvis. See here, this funnel-shaped, like funnel-shaped V. The free end of pelvis has number of cup-like cavities. These are called as major calyxes, single calyx, and minor calyxes. The medulla projects into calyxes as conical processes called renal pyramids or medullary pyramids. The tip of pyramids are called renal papillae. The cortex spreads in between medullary pyramids and renal columns are called as columns of Bertini. See here, this is cortex and this pyramid shaped is in medulla, that is in middle part. From here, the filtered urine passes into this major minor calyxes, and this is the duct or space where urine collects. From here, this is funnel shaped ureter. From here, the urine is passed through ureter to down. Each kidney is composed of numerous, nearly 1 million complex tubular structure called nephrons. These are the functional units of kidney. The kidney cell, each cell of kidney is nephron, and each nephron filters fluid to form urine. This nephron is arranged in the kidney such that its filtration, absorption, and excretion through this neuron takes place very easily. The structure of nephron or uriniferous tubule. Each nephron consists of two parts, that is, Malpighian body or renal corpuscles or the renal tubule. Now first we will see Malpighian body or renal corpuscle. Glomerulus along with Bowman's capsule is called as Malpighian body or renal corpus, which filters out large solutes from the blood and delivers small solutes to the renal tubule for modification. Glomerulus is a tuft of thin-walled capillaries formed by the branching of afferent arteriole, a fine branch of renal artery. This is, you can say, this is a nephron head. In this head, there are two tubules. One is going inside, one is going outside. From here, the blood containing all the waste products come inside this head. And from here, major solutes are taken inside to filter, and other filtered blood is thrown out or given out through this efferent arteriole. Glomerulus is a thin-walled capillaries. What is glomerulus? It is a thin-walled capillaries, like you had seen in alveoli, formed by branching of afferent arteriole, a fine branch of renal artery. This renal artery gives fine branches through which, like alveoli, there is filtration takes place. The afferent arteriole is short and wide that supplies blood to the glomerulus, while the efferent arteriole is a narrow and long, carrying blood away from the glomerulus. Again we will discuss the parts: afferent arteriole, branch of renal artery. Efferent arteriole which goes out of glomerulus. Glomerulus is the head of the nephron, and after this the filtrates go down into the proximal convoluted tubule. Here again there is big nephron we will see. Difference between afferent arteriole and efferent arteriole. What is afferent and what is efferent? Efferent, these are fine branches of renal artery which enter in each kidney and arise from dorsal aorta. renal artery के branches है aorta से निकले हैं. Efferent arteriole, these are fine branches of renal vein which leaves the kidney को छोड़ते हैं and joins with inferior vena cava. This is aorta, this is inferior vena cava. Aorta, pure blood. Inferior vena cava, impure blood. They bring arterial blood to the renal corpuscles. Afferent brings arterial blood to renal corpuscles, and efferent, they carry venous blood away from the renal corpuscles. Afferent diameter is more, efferent diameter is comparatively less. Afferent blood flowing in it is rich in waste products. Efferent, the blood flowing in it is poor in waste products. Now the Bowman's capsule or glomerular capsule is a double-walled cup-like structure that surrounds the glomerulus. The outer parietal wall of glomerulus is composed of flattened squamous cells, and inner visceral wall is composed of a special type of cells, flattened cells called podocytes. This glomerular capsule, outer is parietal, inner is podocytes, or you can say squamous and flat cells. This is the nephron structure. Glomerulus, then from here proximal convoluted tubule. See, this is glomerulus, proximal convoluted tubule. This is descending limb. Again, this is ascending limb. Then distal convoluted tubule, and this is collecting duct. Now we will see renal tubules. These are attached to each Bowman's capsule. It is a long, thin tubule with 3 distinct regions. These regions are described as follows. First is proximal convoluted tubule. Behind the neck, it makes highly coiled network. This. And is restricted to the cortical region of the kidney. Then the Henle's loop. Henle's loop is like tubular structure, ascending and descending. It is quite narrow and U-shaped or hairpin shaped, called as descending limb, that ends into medulla, and an ascending limb that extends back from the medulla into the cortex. It has 2 limbs, ascending and descending. Difference between descending limb and ascending limb of Henle's loop. Descending limb and ascending limb. Descending is very thin, ascending is thick. Descending, direction of fluid is downwards, in ascending it is upwards. Descending limb permeable to water and ascending limb impermeable to water. Why so? Because they have to absorb nutrients or electrolytes which are necessary. Again, this urine which has come into glomerulus, again it is reabsorbed. That means which is necessary electrolytes are reabsorbed in descending limb, and then it is sent to collecting duct. Distal convoluted tubule, DCT, is thicker and highly coiled structure situated in the cortex like the proximal tubule. The coils of both convoluted tubules are intermingled. Intermingled means collecting tubule. The last part of nephron is called collecting or straight tubule, which is lined by cuboidal cells. Collecting tubule of many nephrons open into a large collecting duct. several adjacent collecting tubules converge into a short and thick duct of Bellini. All ducts of Bellini then open into renal pelvis through medullary pyramids in the calyxes. The efferent arteriole divides in to form the peritubular capillary network PTCN around the proximal and distal convoluted tubules of the nephrons. From the peritubular capillary network arise the capillaries of vasa recta, which extend parallel to the loops of Henle and the collecting ducts in the medulla. Likewise, from glomerulus, then proximal convoluted tubule, then descending limb, ascending limb, distal convoluted tubule, and then collecting duct. Now we will see type of nephrons. Based on location in the kidney, nephrons are of two types, cortical and juxtamedullary. Corticals, in majority of nephrons, the loop of Henle is short and extends only very little into the medulla. That means in medulla, they extend very little. They are more in cortex or cortical side, lie in the renal cortex. Such nephrons are called as cortical nephrons. Vasa recta is absent or highly reduced in cortical nephrons. Then comes juxtamedullary nephrons. In some of the nephrons, the loop of Henle is very long and runs deep into the medulla. These nephrons are called as juxtamedullary nephrons. The cortical nephrons forms about 80% of the total nephron count, while rest 20% are juxtamedullary nephron. Cortical 80 and juxta 20. What are functions of kidney? Four functions: regulation of water and electrolyte balance, then regulation of arterial pressure, excretion of metabolic waste and foreign chemicals, and secretion of hormones like renin. There is renin-angiotensin mechanism. When there is fluctuation of BP or BP rises, this renin-angiotensin mechanism activates and they secrete more of urine to reduce arterial blood pressure. Now the ureters. The pelvis of each kidney is continued as a ureter and merges out as hilus. It is a long and muscular tube. The ureters of both sides extend posteriorly and open into the urinary bladder. Urinary bladder. It is thin-walled, pear-shaped, white, transparent sac present in the pelvic cavity. This urinary bladder temporarily stores the urine. It acts like a bag of urine. Then comes urethra. It is a membranous tube which conducts urine to the exterior. Exterior means outside of the body. The urethral sphincters keep the urethra closed except during voiding of urine. When you do not want to urinate, these sphincters are closed, and when you want to urinate, these sphincters open.